Hello everybody, I hope you guys are doing well and enjoying your guys' stem cell class. And also I want to thank Joey for allowing me to talk to you guys. My name is Energy Kier and currently I am an intern at Dr. Prue Talbot's lab at University of California as the CIRM intern. As for Dr. Prue Talbot's lab, we actually study the reproductive biology and developmental toxicology using mammalian systems. Our long term goal is to use human embryonic stem cells to prevent diseases. Much of our current work is done using mouse and human embryonic stem cells to model the pre and post implantation stages of development. Human embryonic stem cells are difficult to use in toxicological assays. Therefore, we are actually developing a new platform using state-of-the-art equipment to study morphological and molecular endpoints in video data collected from stem cells during various experimental conditions. As a part of our lab, we actually study e-cigarette constituents. And before I explain to you guys why we are interested in e-cigarette constituents and what we look for them, I'll talk to you guys a little bit about what exactly e-cigarettes are. E-cigarettes are battery-powered devices that people use to heat liquid into a vape that can be inhaled. E-cigarettes produce aerosols by heating a liquid that can usually contain nicotine. They are ultra-fine particles that can be inhaled deep in the lungs. Therefore, flavors such as diacetyl, menthol, maltol, and others that are chemicals linked to serious diseases. There are other components such as cancer-causing chemicals, heavy metals such as nickel, tin, and lead. These such things are health dangers for pregnant women and developing babies and can cause a developing baby's brain damage and lung as well. The Talbot Lab is interested in understanding how there are pregnant women that are vaping or not vaping that are getting exposed to vape aerosol products that can affect the development of the embryo. Specifically, we are interested in the chemical methylglyoxal because it is a lot more toxic compared to anything that has been seen or tested currently. This chemical is more important in the nervous system. Therefore, methylglyoxal is a aldehyde which is highly cytotoxic and reactive. It is cell permeable and a byproduct of many metabolic pathways. Most of the methylglyoxal is actually generated in our cells that form as a byproduct of glycolysis. Because methylglyoxal is toxic, glycosidic byproducts, and it has high levels that need to be regulated in the cell. That accumulation of methylglyoxal can actually result in apoptosis. And therefore, this chemical is very much important in our lab. In the long run, we want to get from the H9 cells that we use in culture that come from a female line to neuroembryonic stem cells to differentiating neurons. Some of the questions that our lab actually wants to address are the following. How are the vape aerosol particles affecting the human prenatal stages? How are the harmful chemicals and metals like zinc having an effect on the rate of differentiation? And later in development, how is cell death differing in the three primitive germ layers? For instance, the ectoderm, mesoderm, and endoderm. As for some of our experimental approaches, we want to get the primitive three germ layers, known as the ectoderm, mesoderm, and endoderm, using the trilineage differentiation kit. And we want to look at whether the pluripotent embryonic stem cells can even make the different germ layers. And in order to see if we have the three germ layers, we will be doing a fixation process and immunocytochemistry that we will use two antibody markers for each lineage to look at the rate of differentiation and see what markers work. And we'll also use a biostation CT cell culture observation that will view morphological changes happening over time. As a part of our lab, we do cell culturing, and to understand the rate of differentiation, H9 cells, which come from a female line, are cultured and plated in three different 24-well plates with media from the trilineage differentiation kit. With the usage of the differentiation kit, the cells develop the primitive three germ layers, similarly in gastrulation. H9 cells are cultured for 7 days without causing stress on the cells, splitting them, and after are analyzed for effects of e-cigarette constituents on kinetics and efficiency on differentiation. Another experimental approach that is conducted, as I have mentioned before, is immunocytochemistry. And we can use a Nikon fluorescence microscope to view the validation of the primitive three germ layers. Two fluorescence labeled antibody markers for each lineage, as we can see the following. For ectoderm, we can use PAC6 and Nestin, and for mesoderm, we use BRAC-T and CXCR4, and for endoderm, we can use SOX17 and FOXA2. Another very interesting approach that is being taken is using the BioStation CT Nikon microscope. And once the primitive three germ layers are obtained, the BioStation has the ability to obtain time-lapse images over days. These images are collected on a duration of 7 days for ectoderm and 5 days for mesoderm and endoderm. With the usage of the biostation, the morphological changes can be seen very closely, which is very useful when picking time points and checking other assumptions for experimental assays. As for some of the outcomes from the project that I'm working on, it provides insight on how germ layer specification may be affected in vivo. The primitive germ layers will show respectively when labeled with the correct antibodies, and BioStation CT cell culture observation will show time-lapse images representing morphological changes throughout differentiation as well as other features that can be observed. Depending on the immunostaining at different time points, it will show when the pluripotency markers are lost and gained, markers being shifted, and doses of chemicals affecting the rate of differentiation. With this project, I hope to see how such chemicals that are harmful in e-cigarettes are affecting developing fetuses and how it's affecting the mother as well. I want to thank everybody for listening to me today and also giving me the chance to talk to you guys about how the stem cell internship has actually changed a lot of my perspectives and how I am able to actually work with stem cells on a daily basis and conduct experiments and what I've been doing so far as well. Thank you so much and have a great day.